Welcome to this lesson, Introduction to Clustering. The objectives of this lesson are as follows. We will first identify the goals of clustering. Then we will look at our first clustering algorithm called k-means. Then we will describe hierarchical clustering with heat maps. We will describe Gaussian mixture model clustering. And finally, we discuss principal components analysis as a clustering algorithm. In this first section, we will identify the goals of clustering. Recall the difference between supervised and unsupervised learning. In supervised learning, we have features, say x1, x2, up to xp, that the algorithm uses to predict a target, say y. In unsupervised algorithms, we do not have a target associated with the features, or more generally, we do not use any targets or labels in unsupervised training, even if we have them. Unsupervised training is usually equivalent to clustering, the topic of this lesson. Where classification of data is supervised technique, which uses the input features to classify each sample in one of a fixed number of classes. Clustering is an unsupervised technique, which uses the input features to form groups or clusters of similar samples. In this example, in this slide, the same feature space is used for both classification on the left and clustering on the right. On the left, there are apparently three classes in this data set as targets: red circles, green stars, and blue X's. The dashed line represents some decision boundary which a machine learning model has learned to separate those classes into dimensions. On the right, those same samples have been clustered using some algorithm into three clusters delineated by the dashed blue lines. Note that all the samples are colored as blue circles to denote that there is no class association with each sample. They are unlabeled. There are several applications of clustering, three of which are shown on this slide. One application is to determine whether there are what are called batch effects in the data set. A batch effect in RNA sequencing is a technical artifact that manifests when the same experiment is conducted with a certain bias. In this example, there are 7 datasets from GLDS, the old name for OSDR. It is clear that these experiments have batch effects. All the samples from GLDS 168, for example, are in their own isolated cluster, and GLDS 48 and GLDS 47 share a cluster distinct from the others. A second example of a clustering application in space biology research is on the top right of the slide, and it asks the question, which cells are most similar? This particular data is from a single-cell RNA sequencing experiment where individual cells from a tissue are isolated, tagged, and then sequenced, as opposed to bulk RNA sequencing, which is what we see on the left, where the cells are grabbed en masse from a tissue sample. It's clear that the cells distinguish from one another based on their cell type. CD4 from CD8 T cells, megakaryocytes from monocytes, etc. In the bottom and last example, data are observed from different organisms, perhaps phenotype data, and then used to see which species most closely resemble which other species. In this example, eagles and peacocks are most similar, both birds, and lions and bears are most similar. They're both mammals. And spiders and scorpions are most similar as they are creatures with more than 4 legs. As we go up the tree, our clustering becomes more generalized, so the birds and the mammals cluster together. Then finally, all the organisms cluster into a single root, which is the animal kingdom. How clustering algorithms work is they use some sort of metric of similarity to determine how close or far 2 data points are. One metric for data that is numeric is Euclidean distance, and clustering seeks to minimize the distance between points in the same cluster, which is their similarity, and at the same time maximize the distance between clusters, which is the dissimilarity between clusters. In this section, we describe the k-means clustering algorithm. This slide is a graphical representation of the k-means algorithm. The algorithm starts at step 1, where the cluster centroids are initialized randomly in the feature space. In step 2, each point is assigned to its closest centroid using that distance metric we just talked about. And in step 3, the centroids, which are the means in the case of k-means, are recalculated as the means of the points which were assigned to it in step 2. Step 4 is really step 2 again, where each point is assigned to its closest centroid. So notice the data points do not move in this algorithm. That wouldn't make sense. What moves in this algorithm is the centroid. This slide represents a slightly more formal description of the algorithm, not really in pseudocode, but more formal than the images in the previous slide. The first step is called initialization and may be performed in a number of ways, but most often as random, i.e. the k-centroids are placed in the n-dimensional space among the data points. The second step is called the assignment step, where the ith set S is constructed of all those data points x sub p, such that the distance xp minus mi is less than the distance xp minus mj for all the j among the centroids. The third step is called the update step, where the ith median is set to the mean value of all the members assigned to it in the previous step. And the mean, like you would think, is derived by adding up all the values xj in that set Si divided by the total number of those points. This algorithm repeats back between the assignment step and the update step until a stop condition is reached. Some possible stop conditions are described in the slide. For example, if the centroids don't move much from one iteration to the next, we can stop the algorithm. Or if the distance of all the points to their centroids is minimized, stop the algorithm. Or if the distance between the centroids is sufficiently large, stop the algorithm. The hyperparameter k in the k-means cluster algorithm is the user's choice. Even when you eyeball the data, as in this slide, it's hard to tell what the right value of k should be. It'd be great if there was some way we could find the ideal k value of k for a given data set programmatically. One such programmatic way to find an optimal value for k is using what is called the elbow method. The way it works is that you run the k-means clustering algorithm again and again, starting with k equals 1 and calculating some metric like the within cluster sum of squares that we mentioned earlier, and finding the point of inflection in the curve that gets traced. That's the elbow. Obviously, as the number of clusters increases, that within cluster sum of squares distance should get smaller and smaller until the number k is equal to the number of samples in the data set. At that point, each sample is a cluster of 1 and the within cluster sum of squares is 0. This is not an exact method. There's nothing necessarily optimum about the elbow, but it's a decent trade-off between a k that is too small and one that is too large. In this particular picture, it looks like the ideal value for k for this particular data set is 3. I mentioned the word hyperparameters in the previous slide. So what are hyperparameters of a model? Those hyperparameters of a model are those configurable parameters that you as a user of the machine learning algorithm tune, and they affect how an algorithm behaves. Again, as in the case of k-means algorithm, one hyperparameter is the value of k. So if we set k equal to 10, for example, we're asking the algorithm to find k-means, or k-centroids, which are each the centroid of a cluster with some number of data points assigned to it. There are other hyperparameters in k-means. For example, how do those k-centroids get initialized? I had mentioned oftentimes they get initialized randomly, but there might be some other informed methods which improve the overall clustering performance. One such initialization method is called the 4G method, and it selects k random points from the data set as the initial centroids. Yet another hyperparameter of k-means is the distance metric. We mentioned that Euclidean distance, and that is used to measure how good the clusters are for the underlying data. And we use Euclidean distance to measure the average intra-cluster distances, which measure how similar points in a cluster are to each other. Another metric is The sum of intercluster distances, which measures how dissimilar points in different clusters are from each other. One issue when using the k-means clustering algorithm is that it's, like the mean, sensitive to outliers. A single data point that is far out from all the other data points can dramatically change the nature of the clustering because the mean includes those extreme values in its calculation. Instead of using the mean, one way to get around this problem is by using the median as a measure of central tendency, because the median is not affected by outliers. Replacing the mean with the median is a simple change in the algorithm, and we call that changed algorithm k-medioids. In this section, we describe hierarchical clustering. Hierarchical clustering creates hierarchies of clusters, like the name suggests, clusters of clusters, to capture similarity and difference between samples in a dataset. Agglomerative clustering and divisive clustering are two approaches to hierarchical clustering, but they differ in how they build the hierarchy of clusters. Agglomerative clustering is the most common approach. In agglomerative clustering, each data point initially represents a single cluster. Then, at each step, the two closest clusters are merged into a single cluster until all the data points belong to one cluster. This process creates what's called a dendrogram, which is a tree-like structure showing the hierarchy of cluster mergers. Agglomerative clustering is also known as bottom-up clustering because it starts with individual data points at the bottom and builds up the hierarchy by merging clusters going up. Divisive clustering, on the other hand, is the opposite of agglomerative clustering. It starts with all the data points belonging to a single cluster called the root cluster, and then it recursively divides the data into smaller clusters until each cluster only contains one data point. This process also generates a dendrogram, but it's built from the top down, as opposed to the bottom up in agglomerative clustering. This flowchart represents agglomerative clustering. We start at the we start at the start node of the chart and proceed to inputting the measured features to the algorithm. Using some metric for similarity, for example, Euclidean distance or cosine similarity, we compute the pairwise distance between each pair of points in the dataset. We start with every point belonging to their own cluster, so if there are 100 data points in the dataset, there are 100 clusters to start with. Then, while the number of clusters is not equal to 1, repeat the following steps. Merge the closest clusters and update the distance matrix between the clusters. When the number of clusters reaches 1, i.e. all the points belong to clusters and all the clusters are clustered into one cluster, the algorithm terminates. A heat map is a two-dimensional grid that associates a sample with a level for a given feature. For example, this slide heat map shows samples horizontally and gene expression vertically. A particular cell represents how much of that gene was expressed for that particular sample, and it is colored according to the legend. A very red score indicates high expression or heat, and a very blue score indicates low expression or cold, thus the moniker heat map. In this example, the first sample, GSM 3932702, seems to have the highest or hottest gene expression for several genes on the bottom left of the heat map. In addition to graphically showing the different levels of expression for different genes in different samples, heat maps also get plotted with hierarchical clusters that show which gene expressions are most similar along the left of the heat map in this example, as well as which samples are most similar along the top of the heat map in this example. Looking at this heat map, it seems that the first sample, GSM 3932072, is least similar to all other samples combined. We can tell this by observing the height of the hierarchical cluster and its highest between the first sample and all the other samples. The genes most similar are seen as the shortest connections on the left side hierarchical cluster. Indeed, those genes seem to be the hottest genes expressed in the gene heat map. In this section, we describe Gaussian mixture model as a method of clustering. Gaussian distributions, also known as normal distributions, are statistical distributions which are parameterized by their mean and variance. Different values of the mean and variance give rise to different shapes of the normal distribution. In this slide, there are 4 normal distributions shown. The blue curve, for example, has a mean of 0 and a variance of 0.2. With the smallest mean of the 4 distributions, it is the most narrow or skinny, and it's centered at x equals 0. By contrast, the green curve has a mean of -2 and a variance of 0.5. So it is shifted to the left of 0 and is a little wider than the blue distribution. The gold distribution is the widest distribution and it has the largest variance of 5.0. The normal distribution is widely used in statistical inference and is often a good approximation of a sample distribution. The Gaussian mixture model approach to clustering approximates the distribution of all the data as the combination of multiple individual Gaussians. Each Gaussian represents a different cluster. In this slide, the red distribution, f of x, is the data set that we're trying to cluster. Just as with k-means and agglomerative clustering, we need to specify the number of clusters that the algorithm should use to approximate the original distribution. Here we've specified 3, and you can see how the mean and variance of each of these 3 distributions sums to the red distribution. It can be viewed as a curve-fitting exercise where we are using some number of normal curves with different means and different variances to approximate a real curve. With these distributions in place, our clusters are in place. Note that a point along the x-axis could belong to multiple of these 3 distributions. The height of the curve defines the probability of the point belonging to that distribution. So for example, at the point x equals 50, it looks like it has a very small probability of belonging to the green distribution, but a much higher probability of belonging to the purple distribution. The algorithm chooses the highest probability as the cluster assignment for each point. In this section, we describe using principal component analysis as a clustering algorithm. Dimensionality reduction is a method to reduce the number of features under consideration in machine learning. Consider our RNA-seq data from the OSD 255 dataset. It has 16 samples and about 23,000 genes. Each gene represents a dimension, and 23,000 is highly dimensional. Not all of those 23,000 genes contribute a so-called signal or valuable information to the building of a machine learning model. For example, if a particular gene has identical expression across all the samples in the cohort, it offers 0 information to distinguish one sample from another, and it could be removed from the dataset prior to training the model. In this example, in this slide, we begin with 2 data points in 3 dimensions, each is a cube. Reducing from 3 dimensions to 2 dimensions flattens those cubes into rectangles. And with one more dimensionality reduction, we go from 2-dimensional rectangles to 1-dimensional line segments. Principal component analysis, or PCA, is a form of dimensionality reduction, which first searches across the feature space for the vector which captures the highest degree of variance. This first vector is called PC1, or principal component 1, and is not necessarily parallel to the original feature axes, in this example, gene 1, gene 2, and gene 3. The algorithm then considers all vectors which are orthogonal or perpendicular to PC1 and chooses the vector which captures the next highest degree of variance in the dataset. This second vector is called PC2 and is also not necessarily parallel to the original feature axes. The algorithm continues to find as many principal components as you've asked for. Yes, the number of principal components is a hyperparameter to the PCA algorithm. In this example, we use PCA to project the 3-dimensional gene expression into 2 dimensions. Then we can colorize the points in 2 dimensions to see if data that cluster together share some common metadata. Like, for example, perhaps the spice flight samples share a cluster and the ground control samples share a different cluster.